everybody doing? Good morning, good afternoon, good evening. This is Ian Coates here, and here we are at week 8. Can you believe we're already at week 8? Unbelievable how fast these semesters fly. But this is a good one. All right, this is where things tend to take off a bit, and a lot of the things there that we've learned so far, you'll see them here in maybe a different context, and that's one of the great things about biology and learning it, from scratch. Is you start to see the development, how the interrelatedness, the cohesiveness. Yeah, it's really interesting. You'll like this one here. As you can see, we're focusing on DNA. Okay, we're going to talk about chromosomes, and you know what I mean. We've kind of done this there before, but we're going to take it a little further there this time. Okay. And we've got some great links here, and I can't toot these enough. Hit this link here. I think this one here is for OpenStax BC, OpenText BC. And you know what I mean? This is where learning should be. Learning should be free. You shouldn't have to pay money for, tuitions. These you learn when and how you want to. And so it's a really I use it there for a lot of my courses, and there's a link there. It's just strictly on DNA, covering this topic unit, but you can jump around, and there's hundreds, thousands of books there that are all written by PhDs who are interested in this program. So yeah, blah blah blah. That's we'll hit that again. But great link, great. So this is week 8. And would you know that when you're talking DNA here, only about 1/10 of 1% of the human genome differs from person to person. So it's just makes you want to you think about inclusivity and the whole idea. We're not really that different, and you can see there. What is that? 1/1000, so 99. You know what I mean? 99.9 is But anyway, let us go on, and away we go, shall we? Okay, good. DNA, genetic vocabulary. Here we've got genetic information. Instructs cells how to construct proteins, okay, stored in the DNA. So the whole idea of the DNA being a library, a housing facility, that's exactly what it is. Whenever we need to make proteins, because we can't store them, we have to continually make them and use them, right? And there's no place to store them. So per se, anyway, other than breaking down old protein, so we need to make more of it. So it means we have to keep going back to the wheelhouse. We have to keep going back to the DNA and coding for that particular stretch of sequence there that will provide us the particular protein there that we need. A gene, and here we're not talking blue jeans, but apparently this is a blue gene, blue font, yes. This is a segment of the DNA that codes for a particular protein, okay? So that is what's copied. Genome is the complete set of genes. All of them all together, the DNA, the genome, okay? Genomic DNA. And the genetic code, method used to translate a sequence of nucleotides of DNA into a sequence of amino acids. All right, that's the whole point. Again, translating a sequence of nucleotides of DNA, ultimately, so we can get a sequence of amino acids. So when we're looking at genetic material, we're looking at information contained in genes that gets passed on to new generation. You know what I mean? Isn't that what it's all about, right? Source of variability among organisms. Clones, they have a higher rate of, well, even you look at for disease. If if a particular disease hits one person and then there's a clone, well, obviously the other person is going to have difficulty. Okay, so that's why we want everybody to be a little different. And to serve as genetic material, molecules must be able to replicate, store information, express information, okay, and allow variation by mutation. Okay, there has to be that element there, Darwinism, where we are always evolving. Okay, and you think about it, the whole idea of storing information in a series of atoms and being able to go back to that information and recall it and stored, and I mean over millions of generations, billions of generations, millions, hundreds of millions of years. It's quite fantastic. It really is. So, the central dogma theory, and what this is the here when we're talking about the transference of information, right? And basically, how in order to make a protein, the information there is stored in the DNA. Okay, so we have to go to the DNA and unwind it, and then we make a flimsy paper bag copy of that gene, okay, or genes of that stretch of DNA that's copied. Now we have a flimsy paper bag copy of that stretch in RNA language, okay, which can, if needed to be, can taken out, and this is the whole point there, taken to a ribosome where it's read, and the appropriate protein in its sequence according to the DNA is all good to go. Everything is good. So the DNA makes the RNA transcription, which makes proteins translation. And this is what we call the central dogma theory. DNA transcription, okay, then you have the messenger RNA, okay, makes its way over to the ribosomes there where we have the rRNA, ribosomal RNA, and then tRNA, which is your transfer, which is involved in the ribosomal complex, and this is the ribosome reads it, and so much for a finale there, eh? And the protein is made through translation, where the mRNA is read like a ticker tape running through the ribosome. The structure of the DNA, here you can't go any farther than Watson and Crick, 1953. They proposed, or the first to propose, the three-dimensional structure of the DNA. And prior to this they really didn't have it down, this whole idea of it being a double helix that we all know and love there today. Charged off there, proposed there the base composition, A to T and C to G, okay, and percentage of C and G does not always equal, okay, A and T, like ever. That was interesting as well. This right here is a really good movie. You want to pause this whole thing and watch this movie, okay? This gets into the whole the Watson and Crick double helix, and you'll see here that the two strands follow each other, but they follow each other at kind of different angles. And you'll see here that there's a short side, and then you'll see here that there's a longer side, and it works that way, and it opposes, and there's lots to go here, so we're kind of walking before we can run kind of a thing. So pause the video and have a look, and we'll just continue on here, okay? So the Watson and Crick model from 1953 here, they proposed DNA as being a double helix, okay? Two anti-parallel strands, okay? So we're talking a 5' and 3' ends. So you'll see here, I don't know if it's showing up here, the whole idea here is that there will be a 5' end, and then if you follow this strand all the way down, there will be the 3' end on the other side. And if this is the 3' end, you follow it all the way down, there will be a 5'. They're opposing. And we'll get more into that as we go because it's kind of important, okay? Because it's all about direction, okay? And, you know, this is not random. You know, this is high level organization, I mean, at the atomic level. And it's quite fascinating, and it's highly organized, and there is a right-handed and a left-handedness. So being able to identify there a 5' and 3' end and its direction, okay, when we're talking about replication, is paramount. Sorry, didn't mean to stress it, but there you go. Okay. Just moving on here, which we'll come back to that there, the whole idea of A, T, C, and G, okay, the 4 base pairs. This whole monster of, you know, this molecule, you just look how enormous it is. And you remember there from the last PowerPoint there, I think we show you the histones again, okay? And yeah, you see they're all wrapped around these histones, and these are called nucleosomes, okay? And it's there is a right-handedness and a left-handedness to this whole thing, okay? So there's a direction is very, very important. And it's all held together by hydrogen bonding, okay? Like it's just an enormous molecule, and the hydrogen bonding keeps it all together. And we'll show you there the intricacies there inside there. You can see them there with the base pairs, okay? The DNA wrapped around histones there, okay, and forms the chromosomes. Codes in opposite direction. So there's your anti-parallel, okay, 5 to 3 and 3 to 3 to 5 prime. Now, this is a great movie. Stop this video here and give this movie a watch. Okay, it goes into the whole detail of the structure, and 5 to 3 prime goes into it. And after you watch it, you'll be great. Okay, not that you're great now. You'll be better. No, you'll have more knowledge. You get it. All right. Moving on here. You'll see the histones that are involved, okay, and you see how the DNA just winds and ends up supercoiling, okay, which ends up leading there to the chromosome itself, okay. Nucleosome, that's the DNA encompassing there the histones, okay. Just more shots of the same thing, okay. Just with some nanometers in there. I don't think I've ever asked maybe I should, the diameter of a histone, 10 nanometers. Interesting. So please stop the movie and watch this movie. It's a good one. Okay, all of these there, you know, they could be dancing around the, you know, the same thing, but you know, as long as you can find a movie there that you gravitate to and it works for you, then my job is done. Okay, that's the only reason. And I apologize, sometimes I get a little repetitive, but this stuff it's pretty heavy stuff. So, and I hope that you're enjoying it. Now, you'll see that just looking at the slide here, these ribbon-like strands represent the sugar phosphate backbone, and the horizontal rungs there depict the nitrogenous base pairs. And you'll see there that here's that minor groove, okay? And you'll see there that that's about 12 angstroms. That's pretty small, okay? And here we've got the major groove there, 22 angstroms. And you'll see there that one complete turn, okay? There's 34. Coming around and around again. So here it's pretty small numbers that we're dealing with. But again, this is DNA, right? So, but you just look at the high level of order. That's such a small molecule, you know. It's just still blows my mind. The major and minor grooves are apparent, and solid vertical line here shows the central axis. And you see how the DNA winds in and around. There's your double helix, okay? If you look over at the B diagram here, this is a detailed view depicting the bases, sugars, phosphates, and hydrogen bonds, okay, of the helix itself, okay. and you can see them in there, the hydrogen bonding in between. Okay, there's your 5 prime, and opposing there is 3 prime. Follow it around, and there's your 3 prime there. Okay, why is that important? We'll get there. Okay, when we actually get into the replication process, right down to it. And then we have this is just a little demonstration of the anti-parallel arrangement of the chains and the horizontal stacking of the bases themselves. There's your hydrogen bonding there in between, and just showing you here. Okay, here is your base. Okay, adenosine, guanine, cytosine, thymine. Okay, this is your base. This is your sugar. All right, and then this is your phosphate. Okay, so the 3 prime will be open. The 5 prime will be closed. You see how it goes there? We'll get into that there in a little bit. Okay. Moving on. So these are the 4 base pairs here. Okay, you've got adenine, thymine. Okay, and you see how they match up. Okay. And then we've got guanine and cytosine, and you'll see how they match up. Okay, so A and T and G and C base pairing provides complementarity, okay, of the 2 strands, and you'll see there A and T double bond, GC trip. Okay, so getting into the whole idea of 5 to 3 prime, how do we count? Why is this important? Okay, and on the previous slides, I think it was this one here, yeah, it was pretty clear. Okay, your 5 prime, and then your 3 prime, okay, and anti-parallel. Now, how do we count? Well, if you see, here's your base, okay, your nucleotide base, okay, thymine or guanine, and you see how they end up counting the carbons, all right? And you start with, the first carbon, which is here, and this is how they count it: 1, 2, 3, 4, 5, and then there's that 5th carbon there that kind of branches out. And you'll see there that the 3 prime end will always have an open side, okay? And the 5 prime will never have an open side. You'll see there that this phosphate here, that's the phosphate backbone. Okay, it's connected, okay, to the 5 prime there to the 3 prime. Okay, but they got and basically what we have here, the significance of all this, is that now we have a direction. You have a right-handed and a left-handed. That's another level of organization. Okay, not and it's great, it really is fun stuff. And again, I took this picture right from OpenText BC. Please have a visit of them. It's all nonprofit. It's all for the greater good. So it's make a revolution and hopefully never have to buy another textbook again. Okay, because any textbook there that you, for the most part, this OpenText BC, or you have OpenStax, OPEN, Open STAX, Stacks. That's another one there. That's I don't know if that's Yale or Harvard or University of Southern California. Like, all these big schools are involved in it. So enough of free text, and hope everything's good. All right. We've seen this slide there, the whole idea of the eukaryotic cell, okay, when it comes to replication. And you remember this whole area in here there, that was interphase, right? And interphase was made up of G1, S, and G2, where G1 is just the, the cell is, quote unquote, newly formed. I want to say born, but it's not. It's newly formed, and it has a period of growth and development there prior to replicating DNA. And then we go into the S phase. This is the period where the DNA replicates, okay, and chromosomal proteins there are duplicated. Everything gets ready to move. And then we have the period after DNA replication, okay, where the cell prepares for division. And then we get into PMAT. You'll remember mitosis, right? Prophase, metaphase, anaphase, telophase. And then followed, we have cytokinesis, okay? So, this is the cell cycle. You remember. You remember there the little pit stops there, the checkpoints. Where were they located? And what happened there if one didn't pass a checkpoint? What happened to it? You remember, do you? So, when we're talking about interphase here, okay, we introduced what G and S and G2, you know, their concepts, but we didn't really go into their names or anything, but we will right now. But this is the period there from cell formation to cell division, okay? When the cell carries out its routine activities and prepares for cell division. During interphase, nuclear material is in an uncondensed chromatin state. Interphase consists of subphases, which include the process of DNA replication. So you'll see these subphases right here. So G1, you always wonder what the G meant, right? Well, it just meant gap. Sorry for the letdown. This is the gap one. Vigorous growth, okay, metabolism, okay, the cell is just going like wildfire, okay, growing bigger, okay, and the cells that permanently cease dividing are said to be in G0 phase. These are the cells there that aren't doing well, and they get the boot, and eventually get the chomp, okay, phagocized. Then we move into the S, which is the synthetic phase, okay. This is where the DNA actually replicates. Then gap 2, this is preparation, okay, prepping there for division here for mitosis, all right. Prior to division, though, okay, the cell makes a copy of DNA. And you see, like the last lecture or so there, we actually went into what went on, okay, physically with the cell dividing and the DNA doubling. But how does the DNA get copied at the atomic level? This is awesome. This is a lot of fun here. But so prior to division, though, cells may copy the DNA. The double-stranded DNA helices unwind, as you see in here, right? What we call there a replication fork. Okay, a point where the strand separates. Okay. Hydrogen bonds break between bases. New nucleotide pairs pair with the exposed bases. So you'll see there that these are just kind of open bases being able to be read. So, you know, the stability is there for them to be broken apart because DNA has evolved such way. Controlled by DNA polymerase, okay, begins by RNA starting replication, okay, by laying down short strands there that act as a primer. This I have better slides here that I'll show you, and there's a good little video afterwards here, but yeah, it all starts there with helicase unwinding the DNA, and I'll show you better slides, and there's an RNA primer template there that's laid down, but we'll show you this there as we go on. There's a great movie. Pause the video and watch this movie. It's a good one. Okay, hopefully there you had a look at it. We're moving on. When our DNA does replicate, okay, we don't make a brand new copy of it. And you'll see here, there's 3 kinds of DNA replication that are out there. Okay, great link there, Khan Academy. There's another one there that is a great educational link. They have millions of ideas and PowerPoints. Great one. Okay. But getting back into this there, the whole idea is that you'll see that there's conservative replication, where this is completely copied, and so there's a new copy and an older copy. And you can, see the advantages. Okay, you got brand new, spanking new DNA, but then you've got this old rusty one. And how many times, okay, if this is going to replicate again, that means that this, as it stands, is going to live for another generation. How many times can that hold, right? Then you have what's called dispersive, okay? And it's kind of a mix and match, kind of like a candy cane, where it's different segments have been almost breached and intertwined. Okay, so there's new segment and then old segment, new segment linking an old segment. And this is ours right here. It's called semi-conservative. Okay, there's a lot of work that goes into this right here. So what we do is this is a eukaryotic, okay, and we make one copy, okay, of one side, and those two go together, and then we make another copy of the other side of the DNA. So there's one older copy and one newer copy intertwined. Okay, so that's us there, semi-conservative. So the end result is two identical daughter DNA molecules formed from original. Okay, during mitotic cell division, one complete copy will be given to these new cells while one is retained in its original. Okay, it's called semi-conservative replication, and you'll see there conservative and dispersive. Okay, and you can see the pros and cons, but I like ours. So when we get into this whole idea of what is exactly going on, now you see you've got that. Remember that I was harping on the idea there that the 5' to 3', and then we've got the 3' to 5'. It's all about direction, okay? And DNA gets replicated there, okay? And you'll see it gets replicated there in the 3 to 5 direction. And so you'll see here that this stretch of DNA, which I'll get into, this is DNA polymerase, and it's moving in this direction, and as helicase unwinds it, this will just continue on straight and until all of it has been replicated for that particular strand, 3 to 5'. But you'll see here that this DNA, this is 3 to 5'. We're moving in the 5 to 3'. DNA doesn't get replicated in that fashion. It only gets replicated in 3 to 5' direction. So what happens here is the DNA gets bent, okay, and the DNA here is replicated in fragments, okay. There's a really, I think the last video there, if not that video, another video coming up will show what's called these Okazaki fragments, okay. And yeah, it's quite interesting for sure. Okay, there's your DNA gyrase, but we'll get more into these there. You'll see the very next slide. So the DNA polymerase attaches to the RNA primer. So first of all, an RNA primer is laid down. Okay, and then now the DNA polymerase 3 can attach and begin adding nucleotides to the new strand. The DNA polymerase synthesizes both new strands at one time. Okay, there's one leading and one lagging. Okay, the DNA polymerase works only in one direction, so leading strand is synthesized continually. Okay, that's on the bottom here. However, because the lagging strand is backwards, it is synthesized discontinually into segments. And you'll see right here, just the whole idea. And you're thinking, oh my gosh, Ian, why didn't you just leave them all no boxes, right? Because like everything is boxed. But anyway, okay, fine. You'll see here that there's your DNA gyrase, gets it all warmed up and ready to go for helicase here, okay? and it's breaking the hydrogen bonds in between that are holding the base pairs together. Remember, A and T had 2, and G and C had 3, okay, which opens it all up. And you'll see here that this is what we call there the sliding clamp loader, okay, and it has these sliding DNA clamps which holds, okay, holds the DNA and carries it through, okay, so that it can be read in this direction and then also in the opposite direction by using the Okazaki fragments, and this being read discontinually, okay, a stretch at a time, and a stretch at a time, and a stretch at a time, and a stretch at a time, okay? So just going through everything here, there's your helicase there, breaks the hydrogen bonds, and you'll see there these single stranded binding proteins here. These come on there to maintain the stability there of the DNA and hold it open, okay? This is your lagging strand, okay, going from 5 to 3, okay? Your RNA primer goes on first, and that's what your DNA polymerase 3 binds onto, changing there to DNase polymerase 1, which then is able there to lay down the nucleotides. Okay? This is your leading strand. That's a great slide, and there's a movie there for you. Okay? Take a pause, read it, watch it, live it. I'm moving on here. This is just another slide. I don't like it as much. It doesn't even have, 5 and 3 prime on here, but, you can kind of make it go. I just wanted to show you there the whole idea of these nucleotides and how they match up. Okay? All right? And you see them there. There's your leading strand. Okay? 3 to 5. This is your 5 to 3. And these are these Okazaki fragments there. All right, and you'll see there that the DNA polymerase is moving in this direction. But really, it's moving like this because the fragment is wrapped around. Okay? There's another movie. There's a great one. Okay? Pause this and have a look. So, DNA replication, when we're talking about the 2 strands of DNA that form the double helix, DNA molecule there are complementary to each other. Each chain is essentially a mirror image, right? The hydrogen bonds that hold the base pairs together are weak bonds and are therefore easy to separate by helicase. Good little question here. What is the complementary strand of this DNA sequence? All right. Recap. You can have a look here. This is all about interesting things there that we just might ask about. Okay, so replication begins at the point of origin. An enzyme called helicase unwinds the DNA. Replication fork, what is that? Single-stranded binding proteins. DNA polymerase, there's 3 and then there's 1. Primase, okay, and there's your RNA primer, okay. Since the 2 DNA strands are anti-parallel, there's your 5 to 3, 3 to 5, and you see the importance, right, because the DNA is being replicated in the 3 to 5 direction, but the 5 to 3 direction is completely flipped around so that the mechanism there is able to maintain the same direction so both strands can be replicated at the same time. Polymerase can add new nucleotides to the 3' end of the new DNA fragment. Okay, there's your DNA polymerase for sure. And the opposite strand, lagging strand. Okazaki fragment, love it. Okay. Each segment of the lagging strand must begin with an RNA primer. Okay, then your DNA polymerase can add your nucleotides. Okay, then each RNA primer is removed and replaced with DNA at the end, followed by DNA ligase. It's an enzyme that covalently links the new segments of DNA after the RNA primer has been removed. and forming phosphodiester bonds, completing the DNA segment. And that's all we have there for week 8. Please have a look at the questions and review. And good luck. Read the chapter, and we'll see you next week there for week 9. Take care and be well.